So today I'm going to talk about developing a multiplex qPCR for the detection and quantification of root knot nematodes that are important to potato production. And so pictured down in the left, you can see the Columbia root knot nematode, or Mollidigyne chitwoodi, which is one of the nematodes that I'll be trying to detect. And so in the Pacific Northwest, we have two dominant root knot nematodes, Mollidigyne hapla and Mollidigyne chitwoodi, with chitwoodi representing more than 60% of all root knot nematodes identified in soil samples, which were sent to 5 diagnostic laboratories in the region over a 5-year period. And so root knot nematodes are a huge threat to the Pacific Northwest's more than $1.8 billion potato industry, with chitwoodi being the most damaging. Infestations of this nematode are devastating even when initial densities are low, and that's because chitwoodi can withstand much colder temperatures. So while it's not active in the winter, it can wake up and start its life cycle early in the spring and produce many generations throughout a season. And there's zero tolerance for chitwoodi damage. If a tuber has these pimple-like galls that you can see over here, that potato has to be thrown away. We don't have a use for it. Additionally, if just 6% of harvested tubers show signs of root knot nematode damage, the whole crop can be rejected. And this is because nematodes might be undetected in some of the tubers, but they can continue to cause damage while the potatoes are in storage. So our other root knot nematode of interest is Mollidigyne hapla, and it's not as aggressive on potato, but it does still have the potential to cause damage. You can see that down here. And it has a higher temperature threshold required for development, so its population just can't explode in the same way that a chitwoodi population can, just because its time for development during a season is much shorter. And so here we have the root knot nematode life cycle. A juvenile will develop in egg and then hatch and be in the soil, where it will make its way over to invade root tips. And when it gets to the root tip, it actually reprograms root cells to become like a nematode feeding buffet. And those cells get enlarged and form galls. And then at maturity, the female will lay its eggs on the root surface, and then the cycle starts all over again. And so this is kind of a generic plant picture here, maybe some sort of tree, but in potato, the part that we care about, the part that we want to eat, is the potato tuber, which also grows underground. And so these root knot nematodes will also invade the tubers as well as the roots. And so diagnostic demand is increasing in the Pacific Northwest. From 2012 to 2016, the number of diagnostic tests run by 5 laboratories in the region increased by more than 30%. And so root knot nematodes are the #1 nematode problem for potato growers, and they're the #2 pest problem that potato growers are worried about. And so currently, most diagnostic laboratories identify nematodes based on morphology. And to a trained eye, it might seem obvious which of the nematode tails belong to hapla and which one belongs to chitwoodi. But to a majority of us, and probably to the average lab technician, this can be really difficult and time-consuming. And it's easy for a mistake to be made. And so what we could potentially do is transition into using a molecular diagnostic test like quantitative PCR. And so with morphological diagnostics, your prerequisite for running a test for nematodes is to have expert knowledge of nematode taxonomy. The sensitivity and specificity are low to intermediate, especially at low nematode density. Your time input is high and your resources are low. And while your cost is low, that's significantly increased by the amount of time that you're paying somebody to sit and look at nematodes. Versus a quantitative PCR, you just need basic molecular biology lab techniques to be able to run this assay. And so sensitivity, specificity are high, time and resources. Resources, I guess, are a little bit higher just because you're paying for cost of reagents, but you know, that levels out when you consider how many tests can be run at once, and also the amount of time that it takes to run a test. And so how would we do a qPCR for nematodes? So we would get in a soil sample and use a process called sugar centrifugation to get the nematodes out of the soil. And after a worm lysis step, their DNA would go into our individual reaction tubes, and then we would use what's called a thermocycler to do PCR and be able to tell how many are in there. And so the way that this reaction works is you have your template DNA here, and this is a TACMAN PCR, which is the product that I'll be using. So here we have our forward primer, our probes, and our reverse primer. And during a denaturation step, this template DNA comes apart. Our forward primer finds its complementary strand over here on the 3' end, and our complementary probe makes its way and attaches in the strand down from the forward primer. And then our polymerase can run along this strand and do its polymerization. And what happens here in qPCR, which is different from like regular PCR, is that when the polymerase comes by and hits the probe, it cleaves this fluorophore here. And when the fluorophore is removed from the vicinity of this part over here, which is the quencher, it fluoresces, and our thermocycler can detect the fluorescence. And so after many cycles, we get a buildup of this fluorescence, and we can tell how much is there based on the relative fluorescence. And so you get an output that looks like this up here. And what you see with the separation of these S curves are differences in DNA concentration. And so this line over here means that this sample has amplified faster over fewer cycles. And this line on this side over here has amplified slower. It's taken more cycles for the fluorescence to reach a critical threshold for detection. So then what we can do is derive a standard curve. And so we can say if it takes 25 cycles for the fluorescence to hit the critical threshold, then we know we started out with a lot more DNA, versus if it takes 35 to 40 cycles for the fluorescence to hit a critical detection threshold. We know that it's taken much longer for that fluorescence to build up. We had less DNA. And so what we're hoping is that we can kind of backtrack and figure out, okay, so if we started out with this much DNA, how many nematodes did we have in our soil sample? How many chitwitties were in there, and how many haplas were in there? And so our objective is to develop a robust molecular assay using quantitative PCR to improve nematode diagnostics in the Pacific Northwest. And so we'll do this in 3 steps of developing our qPCR. So the first, we will validate our primer probes. Then we will make sure that those primer probes and the reaction works in multiplex. So those reactions separately will work together. And then we'll compare morphological methods to molecular methods for diagnostics. So these are the primers that I'm planning to start out with. These primers have already been published, and they target the commonly used ITS2 region, which lies between some ribosomal genes. And in general, these ribosomal genes are pretty conserved, but this ITS2 region has a lot of variability. And that's why they're often used in molecular assays for species ID. Another kind of bringing us up to our next step is these primers were developed in the Netherlands and Denmark, so we'll need to make sure that they work on Pacific Northwest species, which is why I've gathered for the Pacific Northwest region diverse species of haplo. We have these in culture right now, and diverse species of chitwoodi as well. So I have 5 populations of hapla from Oregon and Washington. I have 1 population of Maloidogyne chitwoodi race 1, and 3 populations from Oregon, Washington, and Northern California of Maloidogyne chitwoodi race 2. And these races, they just specify different host ranges, but both of these will infect potato. And so on to step 2, we need to make sure that this reaction works in multiplex. And so one of the issues that you run into when you start adding a bunch of primers and reagents to your PCR reaction is you can get inhibition, molecular competition. It makes the reaction more complicated. And so we want to make sure that these will still work when you put them together. Additionally, we do have a 10-degree melting temperature difference on these 2 primers. And so basically that could cause us some problems. But if it doesn't work, we'll go back to the drawing board and we can design our own. And so the other thing that we'll have to do is check for cross reactions with other nematode species. And so we'll use other nematode species that we have in culture in our lab right now. But we'll also just go out to the lawn in front of the building and dig up some samples for some uncharacterized nematodes. And this is important because our soil samples will contain a variety of nematodes, not just root knot nematodes. And so we want to make sure that our primers are specific. And next, we'll want to test the reaction sensitivity. And so we're going to hand pick juvenile nematodes and manually count them to create a density gradient to make sure that we can that our PCR quantification scales up. So basically, can we detect 1 chitwoodi in 10,000 haplas? Alternatively, what happens if we have 10,000 haplas and 10,000 chitwoodies? Is that going to completely overwhelm the reaction? So we want to find the reaction tolerance. And next, we're going to collect diverse soil types. And we just want to make sure that there's no byproducts of the soil which could inhibit our nematode sensitivity. And so what we're planning to do is collect diverse soil types, we'll sterilize them, and then we'll spike them with known quantities of nematodes to check for reaction inhibition. And during nematode extraction and worm lysis, we expect soil remnants to be in there just due to the nature of the process. And we need to make sure that it doesn't affect our nematode count results. And so for our last step, we're going to work with a regional diagnostic lab called AgNema. And so AgNema is going to get in some soil samples just like they always do. But this time they're going to split up the sample, and half is going to be retained at their laboratory and half is going to come to us at the USDA. Then AgNema will perform morphological diagnostics, and I'll perform the molecular diagnostics. Then the results will come back to me at the USDA, and I'll compare the 2 methods based on 4 metrics. So the first is we'll compare the nematode counts by sample per method. We'll also be comparing the time it takes to complete those diagnostic tests. We're going to consider labor costs, and we're going to consider supply costs. And so our expected outcomes for this is a more reliable and robust diagnostic assay, which will be available to growers. We want these results from different diagnostic labs to be standardized. And we also want these diagnostic results to help facilitate integrated pest management decisions. Basically, we're hoping to create a better threshold of whether or not you need to fumigate, what kind of actions you need to take to remedy your nematode situation. And we're hoping that that will affect cost savings, the environmental, and protect the environment from the impacts of nematode control measures. And that'll also be important to go into worker safety as we try to reduce the amount of pesticides that we're using in the field. And so with that, I'd like to acknowledge my funding sources, the Northwest Potato Research Consortium and the ARS Potato Program, as well as the USDA Agricultural Research Service, Oregon State, and I'd also like to acknowledge the support we're getting from AgNema and Washington State University. I'd also like to give a shout out to our lab members, especially Amy, who's been super patient with all of my questions and troubleshooting, and also a shout out to Katie for showing me the ropes of being a grad student. So thank you. I'd love to answer any questions that you have.
Excellent, Michelle. Thank you. Does anybody have questions for Michelle?
I have one. Tom Walpert here. Michelle, very good talk. Thank you. You started your talk, you mentioned sugar centrifugation. I'm just wondering if this is sucrose density centrifugation, or exactly what is that centrifugation technique to separate out the nematodes? Thanks.
Thanks, Tom. That's a good question. So I actually haven't done sugar centrifugation yet. I mean, I started in January, and here we are now in the middle of a pandemic. So from what I can understand is that the sugar helps separate out helps float the nematodes to the top of a like a liquid media in a soil sample. So I guess I don't know like the difference between the sugars or what that is, but it's my understanding that the sugar serves as a way to increase the density of the liquid that's in there, so that the nematodes will more easily float to the top.
I suspect you're right, but thanks a lot. I appreciate it.
There's a question from Jay. Jay, do you have a mic, or do you need me to ask your question? I'm going to go ahead and ask Jay's question. Do you expect differences between soils or between nematode isolates?
Differences in the like amount or the reaction results, basically. So I don't anticipate that the primers will be any less specific depending on what like nematode variety we get in. But that's kind of what we're going to be testing. That's the whole point of doing this whole process, is to make sure that these primers are nice and specific and that they work on a broad range of nematodes that we would collect from the Pacific Northwest. And the soil type is a potential issue too. I don't know like what kind of inhibitors could be present in the soil that might follow the nematodes into my PCR reaction. And so that's the main reason why it's important to make sure that we extend all the tests to that extent.
Okay, we have a question from Susanna. Go ahead, Susanna.
Thank you. Nice talk, Michelle. Since you mentioned that we're in the middle of this pandemic, I was appreciating your explanation of qPCR, and I was wondering if you have utilized that explaining it to other people who might be non-scientists about this whole COVID testing, if that came handy in the circle of your family and friends, because a lot of people don't really know what the testing is. Maybe they do by now, but they don't know the details.
Right. Yeah. I'm hoping my family took notes, or anybody else who's on it. This is how we do the COVID-19 testing. And I was actually hoping that people would have read something about PCR ahead of time, and then that would only be help for, what I'm doing in my experiment now. But having, practiced explaining this process so many times before giving this talk, it's going to be helpful, especially when we, all get back together for the next family holiday, or friends ask me, what do you do? I'm hoping that I have a really good explanation for them through practicing with this talk.
Thank you.
Walt, you have a question?
Yeah, sensitivity with these types of assays is often really dependent on how much sample can you process. Usually sugar centrifugation, unless you have massive centrifuges, is a much, much smaller sample. So are you going to be getting the sample size that you need?
Like in terms of,
well,
like how much volume of soil?
Right.
So I think, so the idea with actually doing this, and this is just from what I've learned so far about current methods of nematode diagnostics, is that you might actually only extract nematodes from, or a lot of soil kits will only let you extract from, say, 250 grams of soil. And we're hoping with sugar centrifugation that will be increased to a full, like 5-gram soil sample. As far as, so are you asking about what would happen if we have too dense of nematodes in the sample, or if the...
When I used to do nematode work, we would start with a 100-gram sample that we would wet sieve. And at least from what I know of, the soil kits that you're talking about, is a 2-gram sample is massive for those to be able to handle. So you're seeing already a 10 to 100-fold decrease in sample size by going to the sugar sampling. How is that going to impact your ability to actually detect what's going on in the field?
Right, so our goal is to, when we extract the nematode community, get as many nematodes as we can into our sample. Because the issue now with morphological diagnostics is that if you have just a few, even on the order of 30 nematodes in your sample, you might not catch them with morphological diagnostics. And so the goal of what we're trying to do is to capture the entire nematode community that would be in that sample, and so that could be included in our reaction. Again, I have not done sugar centrifugation yet, and so I can't speak to what it actually means to try to get every last nematode out of a soil sample, or even what it would look like to do that, other than what I've seen in some videos that I've watched and stuff like that. But I'm hoping to hammer out any of those problems over the course of my PCR development. And I'd also be happy to come to you for questions and help. So I'd love to have your input also.
glad to help you with what we can, but yeah.
Do we have any other questions for Michelle? So Michelle, I guess I have a question for you, a general question. Just you had a question and you alluded to just the parallels between what you do and this project and what we're currently in, COVID-19 and testing. And so, when you're thinking about your project and getting your project off the ground, are there any lessons learned from like our failure to be prepared for testing right now, and what that means and how we need to be prepared for testing and plant pathology, food production, and food safety? Have you kind of taken anything away from that you want to apply to your research?
Yeah, that's absolutely a great point, Joey. Like when it comes to developing a PCR test and you want it to be robust and you want it to work every single time, you have to put in the legwork ahead of time to make sure that the test is running at capacity. And I mean, that might look a little bit different in the medical field where once you already know how to, get virus from your sample and run the protocol, like maybe it's just a matter of developing primers for this specific virus. But it definitely reminds me of how important it is to be ahead of things like that. And that means, funding the research that goes into developing the tests for this ahead of time.
Excellent, thank you. Any other questions? All right, let's all unmute and give Michelle a round of applause.
Great job.
Good job.
Thank you.
All right.